It's a great pleasure for me to introduce today's speaker, Don Canfield, who's a chair of biology at D-IAS. And he's an awesome scientist and a great colleague who's been here at SDU for many years. He works at the interface between geology, microbiology, and evolution. So it's a very interesting interdisciplinary field. He originally graduated as a PhD from Yale in 1988. And then he went on to various short employments in Georgia, in California, in Aarhus, in Bremen, various places, Michigan I learned. So, and then in 1996 he was recruited as professor here to SDU. And it didn't take long, then he was co-director of a big center of excellence funded by the Danish National Research Foundation. And when that was finished, he made his own center that he was main director of. Then he had a ESC advanced grant, and just very recently he obtained a prestigious grant from the Willem Foundation. So he is an extremely successful researcher, at the same time being a wonderful colleague to her around. So with that, Don, I look forward to hear your talk.
Well, thanks so much, Susanna. It's really true about all the moving around I did. There's a good colleague of mine years ago who said, Don, you are a man of great mobility. But now finally settling down into Denmark, that's come to a change somewhat. Okay, so how to start this? You know, we're all just sitting around here and we're doing something very important for ourselves without really even thinking about it. You know, we're all breathing, taking a breath about 20 times a minute or something like that, and we really don't give it much consideration, much of a thought. But this person here is up on the top, very near the top of Mount Everest. Where there, the atmosphere is about a third of what it is here, so there's about a third as much oxygen in the atmosphere. And there, it really matters. There are a few people that try to muscle up without any extra help for breathing. A lot of them don't make it. This person did with a breathing mask. And so oxygen matters, you know, and the amount of oxygen in the atmosphere actually matters. It matters to us, it matters to animals, it matters to the living world that we interface with. And, you know, NASA, I guess for their own reasons, and I don't know how they actually obtained this data, but what they did is they tried to figure out where the limits to oxygen toxicity lie with respect to humans. And, you know, again, I don't know how they got the data, but on the long term, humans need about at least half to two-thirds as much oxygen they have in the atmosphere in order to be able to survive. And there's some difference between those that are acclimated to low oxygen, like that live in mountain conditions, and those that are unacclimated. But the point is we need lots of oxygen in order to survive. So the oxygen in the atmosphere is really important for us and for most of the creatures that we interact with. But if we look at a long-term historical perspective on oxygen, and I'm not going to get into all the details, I'll do a little bit, but not so much. There's been a history of atmospheric oxygen over the course of Earth history, and that history just basically is such that oxygen was very, very, very much lower than present time for the first 2 billion years or so of Earth history. There was an increase of oxygen, which I'm not going to discuss too much, unfortunately, but it's a very interesting story. Oxygen rose to levels which we're going to discuss, but which were probably significantly less than today. Then there's a very interesting period in Earth history about 550, 600 million years ago, where we believe that there was another increase in oxygen. And this increase of oxygen has special significance because it's timed roughly with about the time when animals emerged. And we're going to discuss that relationship between oxygen and animals quite a bit as we go further. So my point is there's a history of atmospheric oxygen. Atmospheric oxygen hasn't always been in the atmosphere in the concentrations that we have now, and that's something that I want to dig into a little bit. And one of the things that I want to try to explain a little bit also is how we know some of this. I'm not going to go into all the details, but this is a river sediment from about 2.7 billion years ago. So if we look back on that graph that I just showed, that's during this period of time here when evidence is that I believe is that oxygen was very, very much lower than today. And you know, if you look at this, you can see, okay, well, there's some rounded pebbles of quartz. That's real common for river sediments. But if you look at this gold stuff here, it's also rounded. It looks very much like the kinds of pebbles that you see in rivers, but it's made from pyrite. And that's very unusual. You don't see that today. And the reason you don't see it today is because pyrite reacts very readily with oxygen. And you can do this yourself. You know, you can go and buy a piece of pyrite at the store and then send it out in your garden. Just see what happens over the course of months or a year. You will see that pyrite rust away. But it doesn't here. And the reason that it doesn't simply is because there's a whole lot less atmospheric oxygen in the atmosphere at this point in time. So this is one very, very visual piece of evidence that the Earth's atmosphere has been dynamic, and that when we go way back in time, there was much less oxygen than there is at present. Well, one of the issues then is when did oxygen rise? Remember I mentioned that great oxidation event, this thing that happened around 2.4 billion years ago. And pieces of evidence like this pyrite that I just mentioned has been important in sort of establishing our understanding of the history of oxygen. But, I mean, how often do you go into the geologic record and find an old riverbed? I mean, these things are pretty uncommon. And the challenge then becomes to try to get good resolution, better attempt resolution on what the history of oxygen has been. And a very good friend of mine, James Farquhar, approached this from a very, very different perspective, and it turns out to be a really important one. And what James did, instead of looking for riverbeds, he looked for ancient mud, called shales, ancient mud deposits. And in shales, you can find pyrite that's there for different reasons than what we talked about before. This pyrite is a biological product of anaerobic mineralization. We don't need to get any details about that. But what he did is he looked at isotopes, and oh God, people really get upset when you start mentioning isotopes. Well, it's not radioactive, it's not dangerous, don't need to worry about that. But isotopes conceptually are very, very difficult sometimes to understand. So what I'm going to try to do is break this down into as simple explanation as possible. And what we're looking at is sulfur isotopes. And there are 4 stable sulfur isotopes in the environment. There's sulfur 32, 33, 34, and 36. And you can measure these things on a mass spectrometer. And what happens is if you have a process, you can have processes that fractionate isotopes such that they preferably take, let's say, sulfur 32 over 34 relative to the natural abundance that's there. That's called fractionation if you prefer one isotope over another. But what's neat is that most of the time, the amount of the fractionation depends on the mass. So it's mass dependent. What that means is if you have a fractionation between 32 and 34, the fractionation between 32 and 33 will be half as big. And the fractionation between 32 and 36 will be twice as big. So it depends on the mass. That's mass dependent fractionation. And when James went back and started looking at these old pyrites, he found that the fractionation wasn't mass dependent. It was mass independent. It didn't really follow the mass. In fact, he was so concerned about this that he thought he'd broken the mass spectrometer, and he was really afraid of bringing it up with his PhD or his postdoc advisor the next day, because he was really going to be in trouble. But then he went and he redid some analyses and figured out he hadn't broken the mass spectrometer. What he did is he found a whole new way of looking at oxygen. So if we look at this graph here, 0 means mass independent. Anything above and beyond 0, 0 means mass dependent. Anything above and beyond 0 means mass independent. And what he found is when you go to this part in Earth history, when we believe atmospheric oxygen was low, there's a whole lot of mass independent fractionation. And when we get after that time, doesn't exist anymore. And James is a really clever guy, and what he figured out is a likely possibility is that you need to come up with processes that are going to give you this mass-independent fractionation. And the one that he landed on, which is still what we believe today, is that this mass-independent fractionation occurs from processes that happen in the atmosphere. So if you imagine back in time, you had volcanoes that were belching out sulfur gases into the atmosphere, particular SO2. UV light interacts with the SO2, and he actually demonstrated in the lab that can produce a mass-independent fractionation similar to what you observe here. Well, UV light is blocked by ozone, so what that means is back in this period of time there wasn't an ozone layer on the Earth. Ozone is produced by the dissociation of Earth, or by the association of oxygen. So what that means is that we have an oxygen barometer. Back in this period of time there wasn't enough oxygen to generate an ozone hole, and this time there is. And subsequent modeling suggests that the oxygen in the atmosphere at this time was something like 100,000 times less than today, or less than that. That's what this data implies. So that's the level that we're talking about. Well, you can flip this around and you can say we have evidence for low oxygen, but do we have evidence for any oxygen? So remember the pyrites and the stream beds said the oxygen was low. James stuff says oxygen was low. But is there any positive evidence for oxygen? And in fact, there is a little bit. And again, it's isotopes, and I don't want to dwell on it too much. This is work that was done by a postdoc of mine, Sean Crow, a few years ago in our lab. And again, it's isotopes, but it's the isotopes of chrome this time. And all you need to know in order to look at this graph is that this is the isotopic composition of chrome in rocks, like volcanic rocks or something like that. And if the isotopic composition of chrome gets lighter, what that means, and this is I should even explain what we're looking at. We're looking at an old soil from 3 billion years ago. So it's an old soil like what we have outside here, except it's been preserved in the geologic record. It's developed on an old piece of basalt, which was erupted 3 billion years ago. And as this soil weathers, what happens is that it loses components during the weathering process. And in this particular case, it's lost heavy chrome. And you lose heavy chrome by oxidizing the chrome. It needs oxygen in order for that to happen. So there's a little bit of oxygen in the atmosphere at this time. You don't need a whole lot for this to happen. So there wasn't a lot, but there was a little bit. And I think that this raises a very, very interesting type of a very interesting interface with biology. So if there was a little bit of oxygen in the atmosphere, where did it likely come from? And you can come up with some abiotic ways of producing oxygen that don't require biology, but the best way to produce oxygen, the way that we decided needed to happen in order to explain this results, was by the development of oxygenic photosynthesis, the photosynthetic process which produces oxygen. And the very, very first organisms that developed that were cyanobacteria. So cyanobacteria, their evolution basically changed the world forever because they produced this byproduct oxygen, which has become the mainstream for most of the life that we know and familiar with. So this evidence that we have from the chromium isotope suggests that cyanobacteria probably had evolved by this time in Earth history. And if you ever get the chance to go to a place called Shark's Bay in Western Australia, I really recommend it because in Shark's Bay they have these biological structures called stromatolites. Here's a picture of them. The stromatolites are made from cyanobacteria trapping and binding sediments and then forming these big structures. And if you really suspend your belief a little bit and you just stand there and you look out towards the ocean from the spot, you can imagine that 3 billion years ago, 2.5 billion years ago, that's what the Earth looked like. That's what it looked like if you're standing at the shore, full of these stromatolites. It's actually kind of a beautiful experience. But you can take this observation a step further, and you can say, okay, we have evidence for a little bit of oxygen it seems that cyanobacteria were around. What kind of other biological life might have been able to survive under these conditions of just a little bit of oxygen? In other words, how much oxygen does life require? That is, life that uses oxygen. Yeah?
So there was water on the planet already, and where did the water come from?
Well, there was water on the planet, and the water, the evidence is that the planet became veneered with water about at least 4.2 billion years ago. And there's two possibilities for water. One is that it was added to the surface through basically comets or something like that after the original bombardment phase of the Earth. And another one is degassing from the mantle. And most evidence seems to suggest it's sort of a plastering on from comets that hit the Earth way back when. But it's a lot. There's been constant presence of water on the Earth since as far back as we can see. So the question is, how much oxygen does life require? That is, the life that uses oxygen. And remember, we're talking about 100,000 times less oxygen than we have present today, and that's nothing. I mean, it's nothing. It's so little oxygen that it's almost hard to imagine, and it's so hard to imagine. It's actually very difficult to measure. And as it turns out, our good colleague Niels Peter Resbeck from Aarhus was also concerned with trying to measure oxygen at these low levels. He came up with an extremely clever way of doing it, and I'll try to explain that to you. This is something called the stock sensor, the switchable trace oxygen sensor. And the problem with measuring low levels of oxygen is, let's say you have an electrode. This is a polarographic electrode, so it measures oxygen through oxygen reacting with a platinum electrode generating a current. You measure the current. So let's say you have a little bit of oxygen there, and let's just say that for one reason or another, the baseline on this measurement just drifts a tiny bit. Are you looking at a signal, or are you looking at some drift in the baseline? You don't know. So you can't really understand what these low levels of oxygen are like unless you have another way of doing it. And what Niels Peter decided, which is so clever, is he put this measuring sensor inside of another sensor, which is essentially identical, except that it has a big wonking cathode here, basically a piece of gold, which he turns on and off periodically at a real regular cadence. So when this cathode is on, what it does is it sucks all the oxygen out of the system. So oxygen can't enter and diffuse in and react with this, so you get an absolute zero. Then you turn it off, oxygen can diffuse in, and it becomes measured again. So even though you might have a little bit of a up and down jumping in your baseline, you can always know where your zero is. And by that method, then what you can do is you can start measuring oxygen concentrations down to the levels that you need in order to ask the question, how much oxygen does life actually require, at least microbial life? So I had a really clever Fulbright student named Dan Stolper who came to the lab a few years ago, and he was interested in this problem. So we decided to look at an organism which is well known, in fact one we got from our colleagues in our sister institute, PMB, an E. coli strain. And those of you who know E. coli say, yeah, but E. coli has an anaerobic metabolism, right? I mean, they can switch on fermentation, all kinds of stuff, and that's true. But if you grow E. coli on glycerol, you force it to be an aerobe. It can't ferment glycerol, at least this strain can't. So we forced E. coli to be an aerobe. We set it into a very, very simple experimental chamber like this, where we have the stock sensor, and we have a loop here, and this loop is a silicone loop where we're constantly flowing in oxygenated water. So there's a constant supply of oxygen to this system. So what we do is we just inoculate the thing with E. coli and let it grow. And the population, of course, is going to grow in relationship to the amount of oxygen that it gets from this loop. So when we do that, what you see is that as the population grows, the concentrations of oxygen drop. Look, the amount of oxygen that you have in water saturated with the atmosphere is about 250 or 300 micromolar. So let's say 250 to 300,000 nanomolar. Now we're down to oxygen levels of 10 to 20 nanomolar. So, I mean, the amount of oxygen, most sensors can't even measure it, but this stock sensor can. And what you see is as the population grows, that's these dots here, as the population grows, the oxygen gets drawn down to actually the sensitivity limit of the stock sensor, which is about 2 nanomolar. So now we're down to oxygen levels, which are that 100,000 times less than we have presently in the atmosphere, and E. coli is still growing as an aerobe. So to oxygen levels that low, we can still get aerobic metabolism in microbes, which I think is pretty incredible. Well, because the stock sensor couldn't actually bring us down to the bottom of it, we couldn't really quite get to the bottom of the problem there because the stock sensor didn't have resolution to really track exactly when E. coli stopped respiring aerobically. We generated a model to try to explore that. So we could fit our data to a model and try to imagine what's happening, and that is that if you're a microbe, you're gaining all of your oxygens by diffusion across the outer membrane, right? So you have this diffusion process, and the rate of the diffusion is going to dictate the rate of growth if oxygen is limiting your metabolism. That makes sense, right? So what you can do is you can say, okay, with different sizes, different diameters, you have different surface area to volume ratios. You have a higher or lower surface area to volume ratio when you have bigger cells and a larger one. You have smaller cells. And then you make some assumptions about, well, how much metabolism is converted into biomass and growth. So you have some kind of a growth yield. And that's pretty simple when you do that to be able to say, okay, well, how, at different cell sizes, how much time or how much oxygen does it take to double in a particular time? So this calculation is if you have a 4 micrometer cell, if you have a doubling time of 30 days, you need 3.2 nanomolar oxygen. But if you're a 1 micrometer cell, you can double in that same time with 200 picomolar of oxygen. So now you have this question, well, what is kind of an environmentally reasonable doubling time for organisms in nature? And we generally think of something in this range, like 15 days, as being sort of environmentally reasonable doubling time for nature. If that's true, and you have a standard microbial cell size of about a micrometer, you can see that you can still have a viable population down to 0.4 nanomolar, 400 picomolar of oxygen. So you really hardly takes any oxygen at all for an aerobe to be able to be an aerobe. So you can imagine that even back in this early Earth system we're talking about, when oxygen level was low, cyanobacteria had evolved, but we had an active aerobic ecosystem, even so, which I think is quite interesting. Now we're going to move forward in time. So we've been talking about what's going on back here, and what I want to bring you up now as to what's happening here, because this is going to introduce us into what's happening around the time when animals are evolving. And this is where the geology comes in, and this is where I put on a geology hat and go out and try to figure out what's going on. And I've been so lucky to be associated with some very, very good colleagues in China, where we have access to something called the Zhaomaling Formation. It's 1.4 billion years old. So if we put it in this context, it's here on our graph, about here. And you can see that, the uncertainties in how much oxygen that we have in the atmosphere, at least based on what we knew when I made this graph, is huge, right? We're talking about within a few orders of magnitude, you know it, but that's about as good. So the Zhaomaling Formation, if you can see from these pictures, is actually quite beautiful to look at, and I'm not going to go into too many details except to say that this part here is the part we're going to focus on. It's called Unit 3, and one of the interesting things about Unit 3 is that it actually shows evidence for climate influence on sedimentation, so we can go into Unit 3 and we can extract a signal which is consistent with Michaelis climate cycling, if anybody knows anything about that. It's really pretty cool. That's not the story, though, that I want to tell now. The story that I want to tell now is about how we extract environmental information from these rocks and how that environmental information can help us to constrain levels of atmospheric oxygen. Again, this is going to be kind of quick, but one of our tools is to do geochemical analysis. So we go to the rocks, we measure stuff, and one of the things that we measure is trace metals. And without going into a whole lot of gory details, trace metals behave in the environment in relationship to the chemistry of the environment. So if you have an anoxic environment like Maya, if you have the Black Sea, they behave one way. If you have an oxygenated environment like the Canfield, they behave another way. And when we look at these trace metal signals here, we look at vanadium, molybdenum, and uranium in particular, we can see that they have a very, very unusual behavior. And this behavior is consistent with, in fact, the only place we find anything like this in modern environment is, I'll say it and then I'll explain it, the sediment deposited under an oxygen minimum zone. Everybody got that? Anybody get that? So if you go to, let's say, the coast of Chile or Peru, you find areas in the water column that have no oxygen in them. They're called oxygen minimum zones because there's oxygen above in the water column and there's oxygen below in the water column. So there's this area without oxygen. And if you do the analyses of sediments through this oxygen minimum zone, the oxygen, where there's oxygen, where there's no oxygen, you get one kind of a signal. When you go just below where the oxygen starts to accumulate again, you get another kind of a signal, and that signal looks just like this. So that analogy puts us, I think, pretty securely in that kind of environment. The other thing which is kind of neat is that these biomarkers here are biomarkers for a particular type of a phototrophic organism called an anoxygenic photosynthetic bacteria. And these guys are photosynthetic organisms, but they don't produce oxygen. What they do is they take stuff like iron and hydrogen sulfide and they produce stuff like rust and elemental sulfur. They're perfectly good photosynthetic organisms, they just don't produce oxygen. In fact, these would probably be the first type of photosynthetic organisms on the Earth. So we have an environment that they look a little like that. We have an environment that looks something like this, where we have an oxygenated water column up here, oxygen down here, we have an anoxic sliver of water here, which contains these anoxygenic phototrophic bacteria, and the core that we're looking at is here. That's where it is. And you know, this all of a sudden becomes really, really interesting because you have oxygenated water up here, you have oxygenated water down here. You can have, if you have an environment like this, is there some way maybe you can constrain how much oxygen there was in the atmosphere? Because it's the oxygen in the atmosphere that's ultimately supplying the water down here at depth and supplying the oxygen that we have down there. And we think that the answer is yes. And I'm not going to go into, again, the details, but we can construct a very simple carbon model. And the idea is that you have primary production in the surface waters of the ocean. It's producing carbon. That carbon sinks, it decomposes. It's an oxygen sink. It's removing oxygen the whole time now as this organic matter is falling. In fact, that's what's giving us this anoxic zone here. But as it gets further and further down, there's less and less reactive organic matter, so the oxygen sink decreases. We have a supply of oxygen from surface waters. Basically what we can do is we can come up with a variety of modeling scenarios to try to constrain the amount of oxygen that was there. We need to know how fast the particles settle. We need to know how rapid the primary production was. But we have some constraints on these things, and when we draw a box with this particular model scenario, we can be pretty confident that those results mean that we had oxygen concentrations that were between 10 and 50% of present day levels as a minimum. We open up the model parameters a little bit further in order to be as conservative as possible, and we take these a bunch of these graphs and then we project them in a different way. And you don't need to think about it except that this is the active area here. And if we take this active area here and we just look at the minimum because we're trying to be conservative as possible, we can see that this data constrains minimum atmospheric oxygen to about 4% of what we have in the atmosphere today. Other lines of evidence suggest it was much less than today. These lines of evidence suggest that it was a minimum 4% of today. Okay, so at least 1.4 billion years ago, there was about 4% of atmospheric oxygen in the atmosphere. And now, we're going to tie this back into animals. So, you know, we need a lot of oxygen. Most animals need a lot of oxygen. Yeah?
Is that 4% or 21% or?
It's 4% of 21%. Exactly, not 4% of 100. Yep, good point. So, we need oxygen. You know, most animals we're aware of need a lot of oxygen, but you can sort of ask this question, how much oxygen do animals actually really need, you know, sort of as a minimum? I mean, because what we're trying to understand if there's any relationship between the evolution of animal life and oxygen history in the atmosphere. That's been a prejudice, that's been a hypothesis that's been forever and ever and ever in the literature, that animals evolve when oxygen goes to a point where animals could have a sustainable metabolism. Does that make sense? There's a hypothesis. Well, if that's true, then this 4% is kind of a target, right? In other words, is 4% oxygen enough oxygen for animals? And we don't need to go into that. In order to look at that, we need to look a little tiny bit at the history of animal evolution, at least in a really brief way. So animals evolved from a group of flagellates known as cyanoflagellates, or the sister group to animals. And if one was to think about how animals might have evolved, it could have been something like this, where animals were a cyanoflagellate developed the possibility of becoming a colonial group of organisms, and these colonial group of organisms then through time developed animal attributes of cell differentiation, coordination. In this case, I'm sort of drawing, or this is actually Dan Mills that did this diagram, of which is something very much like a sponge. And we'll get back to why we're interested in sponges in just a moment. But this is one way you can kind of view the pathway. The variety of pieces of evidence that suggest that happened here between about 600 and 800 million years ago, so that's kind of late actually in Earth's history. And we want to try to understand this relationship between the evolution of animals and the available oxygen. So the reason that sponges are kind of interesting is that many, not all, but many trees of animal life put sponges, the periphery, as the most basal metazoan lineage. Ctenophores are sometimes put there too. These two together represent what most people would believe would be the most ancient animal lineages, but the periphery are ones that we're going to focus on now. And there's good reasons to do that too, because the fossil record 600 million years ago has left us with some sponge fossils. So the peripheral sponges not only appear to have evolved early represent the primitive animals, but there's a fossil record of them too. So this actually makes a pretty attractive target kind of a group of organisms in order to explore with respect to their oxygen capabilities for respiration. And I mean, there's only so much you can say from an ancient fossil, right? But I mean, it looks like a sponge. I mean, it looks like sponges that we know from today. So at least in terms of gross morphology, you know, there's still lots of sponge attributes that we recognize even in a 600 million year old fossil. So this one's not 600 million years old. This is just freshly collected from Canfield. And actually, sponges, they're very simple animals in many ways, but they're very fussy. They're not that easy actually to hold in captivity. You wouldn't really think that's true, but they're not. And we tried to get a hold of sponges in a variety of different ways from various pet shops and other places, and finally we got frustrated because we couldn't get sponges that survived, and we just went out to Catamina and got a good supply of this Anaerobic Metabolism, which we decided to explore, and the point was to look at the low oxygen capability of this sponge. So we have a setup which is a little bit similar to the one you've seen before, where we have an oxygen sensor, but in this case we have an oxygen controller which is controlling the levels of oxygen. So instead of having a constant supply of oxygen through this pumping system, we have a periodic supply of oxygen through this pumping system, which tries to regulate the system to constant oxygen tension. But before we did that, we decided to do just a standard oxygen drawdown experiment. So you basically put the sponge in this thing here, and you just let it draw down oxygen and see what happens. And what happens is that it draws down oxygen at a steady rate until you get down to around here, and then the oxygen consumption bends. And that means that when you get to oxygen levels down around here, around 10% or 15% of air saturation, then the oxygen starts to control the respiration rate of the sponge. And you can generate this, you can make what's called a Michaelis Menten kinetic plot of this data. And if you do that, you find that the sponge halochondria has a Km value of about 5.1% of oxygen levels. In other words, at 5.1% of present day levels, halochondria respires its half its maximum rate. And it's not terribly physiological, but it's something that people often use to try to appreciate where animals, organisms in general, start to become influenced by oxygen and oxygen availability. So this is actually a pretty low number, and it's getting kind of close to that 4% we were talking about. So it appears that halochondria actually is not terribly sensitive to oxygen, except when you get down to pretty low levels. So it seems to be fine with oxygen until it gets pretty low. Well, we tried to explore this in a series of respiration experiments where we wanted to see, well, does halochondria keep respiring? How does it behave over several hours if it kept at very low levels? So we put that circulating system on, and so what happens is this, in the circulating system, as oxygen gets drawn down here to 1% of present day levels, then the oxygen builds up, and then that circulation system stops and oxygen gets drawn down. So we get a series of respiration experiments, and we keep doing this, and we can do it for several hours, we can do it for a day. Halochondria just seems to be respiring happily between about 1 and 4% present day oxygen levels. So it seems like halochondria is more or less satisfied with these kinds of oxygen levels. But I mean, how do you know when a sponge is happy? You can't really ask it, and when they do die, they sort of die in parts and pieces. And it's really kind of hard to appreciate when a sponge is actually doing well or not. So we decided to look at another sponge in order to explore these things in a little bit more detail. We looked at Tethya wilhelma, which is a very interesting sponge. It's a little guy. This is about a centimeter max in size with these silicone spikes. And one of the things that attracted us to Tethya, which we'll see in a minute, is Tethya actually displays some very interesting behavior. And you don't think about sponges maybe as having behavior, but they actually do, and some of it's pretty cool, as we'll see. So with Tethya, we did the same old oxygen drawdown deal that we did with halochondria, and we came up with a Km value which was almost the same. So at least from this sort of gross measure of physiology, Tethya has a very, very similar oxygen tolerance to halochondria. But the thing that really is interesting about Tethya, which made us fascinated by it, was this. Look at this, check this out. This is a video which is taken over a span of about a day. And you can see that Tethia has these periodic contractions. Let's look at that again. An individual contraction takes about half an hour, something like that. I mean, it looks pretty violent, right, when you see it sped up like this, but it actually does take a little while. Contraction takes about half an hour, something like that. So basically, what halochondria is doing is it's squeezing its water canal system. So sponges are made with a series of water canal systems where the water flows in and is filtered by they're called cladocytes within the cell and within the organism, and it's basically squeezing those things shut, and then it's opening back up again and it's squeezing them shut. And that's one of the things that we're going to try to look at here and try to understand. So if we go back to this, what we see is that halochondria has this kind of regular rhythmic beating. And you know, each one of these cycles takes some hours, and it just keeps doing this, keeps doing this, and keeps doing this. So what Dan did is he subjected mitochondria to a variety of different levels of oxygen. So we see we have 100% down to about 2%. And there seems to be some kind of a trend here where the rhythmic contractions decrease with time. But by the time we got to 2% oxygen, stopped doing this. Stopped doing it altogether. It just sort of stayed inflated. So there seems to be some kind of a relationship between the rhythmic contractions that mitochondria has and the presence of oxygen. And one of the reasons that this could be, we don't really know why. We don't really know what that relationship is, but it's interesting because there's a strong relationship with this animal's behavior and oxygen, which is kind of cool anyway. One possible reason, though, is that mitochondria, like us, and like I want to say all other animals, most other animals anyway, has an people like to call it an endosymbiotic. I prefer to call it endobiome. And this biome is made up, we actually don't know exactly what it's made up in the Tethia yet, but it's generally made up of a series of microbes. Many of them are anaerobic. And you could imagine that if this microbiome that sponges have is important to the well-being of the organism, it probably is, then what Tethia could be doing is generating periodic conditions which are favorable for the microbiome. So it contracts, squeezes the oxygen out, gives it a better possibility for the interior of the body of the organism to become anoxic, enabling the microbes, and then when they've done their thing, then it opens up again, and then sort of satisfy the needs of the eukaryotic cells. It's a hypothesis. We don't really know if it's true or not, but it makes sense also in the context when oxygen gets low enough, then it doesn't need to do that anymore. Then it's satisfied the requirements of the internal microbial population, and also apparently its own cells, and then it doesn't need to do this inflation contraction stuff. It's just a hypothesis. It's something we need to test, but it does suggest first that there is a relationship between Tethia behavior and oxygen, and there might actually be some really interesting crosstalk between the microbial population in the Tethia and the eukaryotic part of the organism, which I think is pretty fascinating. The idea that you can get sort of cross the domain of life communication, which is pretty neat. I'm almost finished. There's one other thing I want to bring up in this context. Again, it's hard to know when these organisms are happy, but what Dan did is he grew Tethia at almost full air saturation, and then for a long time, several days, he grew Tethia at 0.2% atmospheric oxygen. I mean, that's really not very much, and it looked happy. I mean, it's not smiling quite, but you can see that there's no signs of necrosis. The color is good. It doesn't contract right because there's not enough oxygen for it to do that. But it looks good. And now the new piece of information that we just got last week is that, like also other animals, like all of them, probably Tethia has an anaerobic metabolism. You know, we do that when we run, for example. You know, parts of our metabolism becomes anaerobic. Well, Tethia also has an anaerobic metabolism, and that metabolism was not switched on even at these levels of oxygen, suggesting that the Tethia actually doesn't really care that much, even when oxygen gets so low, suggesting a very, very low oxygen need by this organism. This is again stuff that we need to get in more detail, but to try to summarize up a little bit, it seems then that at 4% oxygen, there was enough oxygen around in the environment for animals to have evolved long before they actually did. So that means that animals evolved not necessarily in response to increased atmospheric oxygens, but in response to the time it takes to evolve an animal. It just takes time to put all this sort of biochemicals junk together before you can actually create an animal body plan and a functioning animal system. So that's one thing. But this environment, even though, let's say it's 4%, let's say it's 10% atmospheric oxygen, it's still an environment where there's a lot of dynamics in oxygen. You know, a little change in oxygen, let's say in our waters here where there's so much oxygen in the atmosphere, might not matter a lot, but that same amount of change when you have a little bit of oxygen in the water could actually cause oxygen to become very, very low. If you're a sponge, you can't really move out of it. So being adapted to low levels of oxygen like these sponges appear to be could be a real advantage in the environment, even when oxygen was above this sort of 4% threshold that we were talking about. So basically, because I'm running a little bit over time, I'll go straight to thanks. And there's a whole lot of people that have contributed to this work. Dan Mills, Lars, Christian Baum, Emma Hamlin, a whole bunch of people. And of course, your funding agencies have been responsible for funding this. And I thank you for your attention.
So you showed more or less these step functions, and thinking about, like, obviously there's a lot of time in between in the transition phases, but still, do you believe that once you get to today's level of oxygen, that basically during that transition phase it was just an explosion of a few species that managed to produce so much oxygen as waste product, and from there on basically we see all the diversification? Or what is kind of the idea, what gets you from one level of oxygen to the other in relationship to the amount of biomass in the following?
Well, you're kind of asking several questions all at once. So, with respect to the step functions are likely a result of an interplay between biological and physical forces. So without getting in too much detail, oxygen is controlled. Yeah, there's a slide which might help this. The oxygen atmosphere is a kinetic balance between the rate of oxygen production and the rate of oxygen consumption. And oxygen is produced by the burial of organic matter. And actually, it's going to take too long to get into that. But the point is there are feedback mechanisms which help maintain oxygen at a reasonable level. You can still get fluctuations, but you have feedback mechanisms. Now, the biggest, most important biological event in the history of life on Earth probably wasn't the evolution of us, but it was the evolution of cyanobacteria. Because all of a sudden, remember I mentioned before, anoxygenic phototrophic bacteria use iron and sulfide. Where do you find iron and sulfide? And it's around, but it's volcanoes, it's not that abundant. Oxygenic phototrophs use water. Water's everywhere. So it's with the evolution of cyanobacteria, all of a sudden you have this electron donor, which is readily abundant. And I've made calculations, and others have too, that the activity level of biosphere probably increased by 1,000 with the evolution of cyanobacteria. So now we're talking about a whole other world, a whole other place with a whole other prospect for biological cycling, and it's probably been a very, very important drive for evolution. So just not only having the oxygen available for new types of organisms, but just a strict increase in the biomass, the productivity of the earth. And then we can talk about other time intervals, but I think that if I were to focus on one, I'd say that was interesting.
I'm curious about the happiness of the sponges.
Yeah, me too.
So with the bacteria, I would think that it's happy when it's multiplying. So in any way, could you measure cell growth or cell, I don't know, biomass increase
Yes.
of any sort?
We could. So when Dan was looking at mitochondria, he kept it at a constant 2% atmosphere, and what we could see was that it was growing.
All right.
It actually was. I mean, or growing was changing its biomass distribution. You know, we didn't weigh it before and after. We probably should have done it. We could see that there were areas where it actually was having biomass, and it looked nice. I mean, when you see a sponge that looks dead, it's black and it's ugly. You know, when you see a sponge that seems to be happy, it's yellow. So, but you're right. You know, being able, but we're thinking also, you know, through transcriptomics. You know, do we see the expression of stress genes and stuff like that? I mean, that's one of the things that we're trying to move into.
Because I would suspect that at high oxygen levels, you actually will have like an increasing oxygen radicals and other things that would actually probably damage the sponge.
That's a possibility. Yeah.
So I'm more curious is like total carbon content. Does it actually grow as a total organism when it's happy?
Well, it certainly grew in parts,
Yeah.
and then we can get into this interesting discussion Lars and I have all the time about, you know, what defines a spongy animal.
Okay, yeah.
It's kind of interesting, right?
Yeah,
I mean,
yeah.
what are the limits?
Don, do these primitive organisms have efficient ways to capture oxygen?
No, it's all by diffusion across their membranes. So it's not very efficient at all. Well, it actually is efficient in a way,
Yeah.
but it's the same way that microbes do it. I mean, you know, they're basically the same thing. They don't have a blood system, for example, in order to concentrate oxygen in some sort of
No, for, you know, enzymatic systems.
yeah, just diffusion across the membrane.
Yeah, I'm just curious to ask you, do you see at all an analog or a parallel to sort of larger microbial carriers, protists like foraminifera, that we know have an endobiome that enables them to survive in oxygen and low oxygen environments, and how, you know, if you think of the choanoflagellate as the precursor to the sponge animal, that maybe something could be gained by studying some of these protists.
We're doing that actually,
Okay.
but we don't have enough information to be able to intelligently answer your
Okay.
question. But that's a good question.
Yes, sorry, I don't know a whole lot about bacteria, and so it might be a very naive question. So, certainly related to the first question. So, with these sudden changes in oxygen levels, I mean, I know from like mammalian cells that we have a number of genes just there apparently to protect ourselves from oxygen. Is there any evidence, like in fossils, of like a massive extinction of microbial species during these?
No, this is. I had a very interesting telephone conversation with somebody yesterday about this same issue. There's a prejudice kind of in the literature that when oxygen rose, there was a massive extinction of microbes, a generated environment very hostile to the anaerobic microbes that we were before. But when you really think about it, you know, all you need to do is dig in a marine sediment like a millimeter below the surface, and then you have a microbial world which is massive. So you redistributed them. You put the anaerobic microbes in a different place, but you didn't cause an extinction. No way. In fact, if you think about it the way we just described it, the opportunities with more biomass and more production probably actually increased rather than decreased the magnitude even of the anaerobic.
There was a reason I asked you about the percent, and that is that as a physiologist I regard that the oxygen concentration at 100% of atmospheric levels as hyperoxic,
Yep.
potentially dangerous to.
Yes,
And you'd agree?
Yes. I would, absolutely.
So I'm interested in embryonic development and implantation. And there I think we're talking about something like around about, as far as I'm looking at it in my head, around about 25% of atmospheric levels being the normal type of oxygen concentration that these cells thrive in. So it's not very far down from that to the 10%. So my point I'm trying to make is that we haven't become vastly more dependent on oxygen since we evolved from sponges.
Well, that's an interesting point, actually. I guess I would view it as kind of yes and no, because it depends on where in the body we're talking
Yes,
about.
it does.
You know, we still have this requirement that we need to live in an atmosphere with this amount in order to be able to survive. But you're absolutely right. And picking apart just where in the organism, you need oxygen or you don't, I think is really fascinating. And that's something we're going to want to do with the sponges too. We have this developing observational series of observations about how much oxygen they need. But what about inside the sponge? What does that look like? I think that's going to be the next phase of some of our work. We'll try to get into these kinds of questions.
These sponges, are they specialized cells?
Yes.
Yeah. So how different are they?
Yeah, very different. So there's the choanocyte cells, which are responsible for the beating, sort of the ventilation of the sponge, and also some of the initial extraction of the food. And then there's also mesophyll cells, which also can extract larger particles, and they're responsible for actually migrating around in the sponge. And they're a little bit like stem cells, and they can actually develop different types of cell types depending on what they need. But there's nothing like the number of cell types that we have.
Further questions? If not, then I have this little present for you.
Oh,
Yes.
fantastic. Thank you.
And then I'd like to announce that we have little serving upstairs with some coffee and cake and I don't know if there's fruit and other stuff. But please join us upstairs, and please join me. Thank Don once more.